My name is Michael Haag, and I'm CEO for Visano Theranostics. I'm here to talk to you today about something that we've developed, which we're pretty excited about. And so we've developed imaging agents that are real-time, targeted to tumors, and help physicians go after prostate cancer biopsies and enhance that process, as well as focal therapy. So in this room, men, if you're of a certain vintage, right, your doctor's looking at your blood work, and they might say to you, hey, listen, I see your PSA levels are rising, and I want you to go have a prostate cancer biopsy. And we should all groan, right? So you go for that prostate cancer biopsy, and in a lot of cases, you start on this side of the screen. Most places are going to start you with an ultrasound. You go in for your ultrasound. It's fast, it's quick, it's pretty cheap for the hospital, but the doctors are pretty much flying blind. And so you don't get the results that you need to have to go forward. You can't visualize the tumors. In certain circumstances, in certain hospitals, you have the CT and the MRI, which have great accuracy, but they're slow. It might be disjointed in the process, and they have a lot of costs for the hospitals. As a result, up to 50% of the prostate cancers are missed, and at least repeat painful biopsies that are costly. And for the patient, you go home, right? You're thinking you're cancer-free. You don't have to worry about this. Then you get that phone call. We want you to come back. You've got to do this all over again. At Visano, we've developed nanobubbles. So it's a lipid particle with a gas shell that is targeted specifically to the prostate cancer. And it's very simple. It inserts regularly into the workflow, and you just inject, you image, and then you biopsy. And so just focusing on this slide over here in the upper left-hand corner, you see a normal ultrasound, right? That's what you're looking at if you're the physician and you go in to target this biopsy versus our ultrasound. You have a heat map. You see exactly where to place the needle to take the cells to be able to move forward. We have a very defensible position. Our IP came out of Case Western Reserve University and University Hospitals. We have extensive patents in this portfolio. We cover the composition of matter on the bubbles themselves, the way that we target the bubble. In fact, we actually have patents on using these bubbles as a therapeutic. So the same bubble, without me changing anything, can be used. If it goes inside the cell, we can pop the bubble and it'll cause the cells to apoptose, leading to therapeutics. There are multiple targets that we can go after. Today, we're focused on our first product, which is prostate cancer related, but we can go after other soft tissues as well, all with the same chemistry. So how is this problem being solved today? In a number of circumstances, ultrasound is what's being used, but it has the problems I said before. The physicians are flying blind. In the meantime, MRI has stepped in. And MRI has stepped into a process called MRI fusion. And MRI fusion can do a pretty decent job, but it's costly. It takes a different level of expertise, a different level of person required to be able to do this work. It's not available in every center. And so it's also time dependent. An MRI is usually taken either up to a day before, a couple days before, a week before. And so what we'd like to say is that the MRI shows you where the cancer was, but with real-time imaging, Visano can show you who the cancer is. And we can get that MR level accuracy at a fraction of the cost. From the market, if we start all the way out, it's a $1 billion plus market for all the soft tissue imaging that we can go after. Small parts imaging is what they like to call it. So the thyroid, the breast, the testes. We're going to focus on prostate cancer biopsies, which have over 3.4 million of them that are done every year worldwide. If we go just the US, that's a $1 billion market. And if we go just US, that's about a $400 million market. And we think in 5 years, we can capture about 30% of that market for a little over $100 million. What's really exciting to us is that one of our physicians, actually two of our physician advisors, put on our radar this thing called focal therapy. Focal therapy is for folks that have prostate cancer and they have defined lesions, and you go in with either high-frequency ultrasound or cryoablation, and you're trying to target those lesions and preserve the rest of the tissue. Our physicians have said, if you had this for that, we would use it tomorrow. We want to be able to go in there, and so that's a growing market we're keeping our eye on. What's exciting for us, I don't have to change my bubble at all. That same bubble that I'm using for prostate cancer biopsy, I can use for focal therapy. Another key driver for us is that this is going to save hospitals lots of money. We anticipate reducing repeat biopsies will save the system over $2.3 billion a year. So we have a very strong preclinical package. I like to say that our founding team has done as much as they can do preclinically at an academic medical center as possible. We have large animal studies. We see big tumor signal to noise increase showing the imaging. We have no toxicity whatsoever that we've been able to see. This is done all the way up to large animals. And so we think we're ready for GLP tox and GMP manufacturing. And we've been validated by being selected for Ohio TDSF and the Trailblazer Accelerator. Where we are now, we have $250,000 that we've raised that'll get us to a pre-IND meeting. We'll be able to do all our pre-IND work. We're seeking to raise $1.25 million to get us to that IND. This will allow us to do the GLP toxicology, the genotox, and the manufacturing. And we are looking to partner with somebody here to get that match from JumpStart of $225,000 to be added to what we have. Our team is outstanding. One of my founders and one of my advisors is here in this room. Strong regulatory experience taking drugs into the clinic from big pharma. Strong medical experience seeing these patients every day. And we welcome you to join us in our seed round and help to change the lives of men facing this horrible disease and help physicians discover cancers more efficiently. Thank you.
Thank you so much, Michael. Great job. Questions? Yes.
Great presentation, Michael. Thank you. I'm Mark Chance, Cleveland Life Science Advisors. Could you walk me through the regulatory bits a little more? I'd like to hear, after the IND is filed, et cetera.
Yeah, so we are working on a traditional IND pathway. We asked the regulatory consultants if we can do a pathway called the 505(b)(2), which is where you have a drug that looks like it on the market already. That was a no-go. So we have to do an IND. The IND is going to have that full preclinical workup. So we have to do the toxicity, we have to do the PK, we have to do the animal studies, the herd studies. And so that's what we're really looking for, that $1.25 million to round out our round. So that way we can start our clinical trial. And we think that whole process from where we are to then is probably about 18 months.
Thank you.
Thank you, Mark. One more question? Yeah, great.
Michael Gilkey, Theta Tech AI. I'm curious if you're able to develop a MRI equivalent of PIRADS with ultrasound, where you could give more of an interpretation of how bad the prostate cancer could be.
I might want to have my founder answer that on the science side, but for as far as I know, one of the things that we can tell is that the ligand that we're using to target this is based on something called PSMA, and PSMA does tend to be more expressed the more developed the tumor is, but that's not an area that we're focused on initially. We're going to go for a very simple FDA approval of margin identification or structure identification to make this as broadly applicable as possible.
Thank you. One more round of applause for Michael. Great job, Mike.